will be recorded. It will be recorded and archived along with past and future webinars on our website. Today's webinar focuses on the impacts that invasive mussels pose to native fisheries in the Great Lakes. We're pleased to have representatives from the University of Minnesota Aquatic Invasive Species Research Center, Trent University, and the Michigan Department of Natural Resources joining us today to share their experiences. A few technical and housekeeping items before we go on. We always strive to minimize technological issues during these webinars, so we apologize for any issues that may arise, but we have a few logistical items that might help address this. Audio for this webinar is automatically streamed through your computer speakers once you join the Skype event. You also have the option to dial into the webinar using a telephone. To change your audio to a phone line, select Switch Audio to My Phone from the banner at the bottom of the screen and click Connect from the pop-up window. A second pop-up window will list the number to call into as well as the conference ID number to connect to the webinar. In order to minimize the potential for interruptions and background noise, we have automatically muted all attendees. Attendees will remain muted throughout the webinar. We do encourage you and would like to accept written questions from attendees during the webinar. So if you have a question, you can type it into the chat box, which is signified by the chat bubble icon in the lower left-hand corner of your webinar screen. Questions may be submitted at any time during the webinar, and depending on time, we may answer one or two questions following each presentation. The majority of questions will be addressed after all the presentations have been given. We do our best to take all questions or comments during the webinar, but if you need to leave the webinar before we've gotten to your question, we encourage you to review the recording that will be posted on our website to hear your question and answer. If you're experiencing technical difficulties, you may send a message to the chat box as well. To view presentations in a full screen format, you can change your screen layout to Content View. To change the layout, select the layout icon in the upper right corner of your screen, indicated by the red circle on the slide, and choose Presentation View or Content View. So hopefully those items are helpful to you for today's webinar. And we are now going to move to our presentations. So I'm pleased to welcome our first presenter this morning, Megan McEachran. Megan McEachran is a graduate research fellow at the Minnesota Aquatic Invasive Species Research Center. Her research on zebra mussels as a biology major at the University of St. Thomas inspired her to pursue a PhD in conservation sciences at the University of Minnesota. There she is studying the risk of fish pathogen and AIS introduction via the live bait fish pathway. So Meg, we have handed off presenter status to you, and you should be off mute or unmute yourself, and we should be ready to go. Meg, are you able to unmute your phone line?
Are you able to hear me now?
Yes, we can. Perfect.
Are you able to hear me all right?
Yes.
Okay, great. Well, thank you very much for having me here today. I'm really excited to present this work that I did as an undergraduate at the University of St. Thomas, where we use stable isotopes to examine the effects of zebra mussels in an invaded lake on the broader fish and invertebrate community. And I always like to acknowledge the University of St. Thomas, where I did this work, but also our partnership with the Minnesota DNR. We had a fantastic set of collaborators who helped with the field work as well as analysis and writing of the manuscript. And of course, our funders, the Legislative Citizen Commission on Minnesota Resources and the Environment and Natural Resources Trust Fund. And I was just one of many undergrads to be lucky enough to work on this project. There were several others in my lab. And I also like to give a shout out to my advisor, Dr. Kyle Zimmer at the University of St. Thomas, and Brian Herwig, who is our DNR collaborator, without whom this would have been impossible. So most people on this webinar are probably fairly familiar with how zebra mussels affect water quality. They are filter feeders, therefore they decrease cestin or free-floating algae in the water column and increase their clarity. But it's a little bit less clear what this means for the overall fish community. So the way we wanted to take a look at this is examining two lakes in, yeah.
Sorry, we need you to click the takeover presenter status at the top of your screen so we can see your slide sharing.
Sounds good. Okay, how does that? Great.
It looks great. Thank you.
Thank you. So we examined two lakes in northwestern Minnesota, Lake Carlos, which was invaded by zebra mussels in about 2009, and Lake Ida, which was not invaded by zebra mussels until very late towards the end of our sampling. We actually found the first zebra mussel in that lake. These lakes are in the same watershed and are of very similar area and depth, and therefore they provide an excellent opportunity to kind of substitute a space-time study designed to examine the effects of zebra mussels. So we wanted to know, how do the fish communities in Lake Carlos differ from the fish communities in Lake Ida, which up until the point of the study did not have zebra mussels in it? And as you can see in the graph on the right, the Secchi depth transparency, or the water clarity, greatly increases after the onset of zebra mussels, illustrated by the arrows. But we want to know, what does this mean for the broader fish communities? So first, we wanted to see how the presence of zebra mussels affected reliance on littoral or nearshore energy sources. Also, how does the presence of zebra mussels affect the trophic levels of fish consumers, where they're feeding in the food web? And finally, what is the implications of both of these potential changes for fish isotopic niches and the fish community? So because we're using stable isotopes, which I'll explain in a minute, we needed to take a lot of tissue samples because tissue of the organism contains a certain isotopic signature from which you can derive information about where and how it's eating in the food web. So we did a ton of sampling and a lot of collaborative effort with the Minnesota DNR to sample several fish and invertebrate species in the two lakes over 2 summers. And then we sent them off for stable isotope analysis at the University of California, Davis, where they did the actual isotope analysis. And for those that aren't familiar with stable isotopes, I wanted to take just a bit to explain how they're used in ecological, especially aquatic systems, and how we're able to make the interpretations that we are based on stable isotope signatures. So the two most commonly used stable isotopes in ecological studies are carbon and nitrogen. Carbon-13 is the heavier isotope of carbon. It's less common than carbon-12, and it occurs in the environment in predictable ratios. So by understanding the isotopic signature of an organism, you can understand how and where it's getting its food. So lower or depleted carbon-13 indicates that an organism is coming from a pelagic area. As opposed to an enriched or a higher 13C signature, which indicates more reliance on littoral or nearshore sources. This is because phytoplankton floating in the water column has its pick in terms of what kind of carbon it wants to take into its tissues. Organisms typically select against heavier carbon. They don't like the carbon-13. It's harder to work with in their tissues, so they select against it. So if they have the choice, they'll take up the lighter carbon-12. However, organisms trapped in the littoral zone have less of a chance to select against carbon because they have what's called a boundary layer effect of the water actually passing over the surface of their tissues, which limits the amount of carbon that they have a choice to take in. So they have, as a result, a higher 13C signature. And because carbon has very low fractionation up the food web, by understanding what an organism's carbon-13 signature is, you can actually calculate its reliance on proportion littoral energy using this model, which is from the Post paper 2002. So all you have to have is the known trophic fractionation of carbon, which is something like 0.39 per thousand, so it's very low, the 13C signature of the baseline, and the 13C signature of your organism. You can also use nitrogen isotopes to study energy flow. Similarly to carbon, N15 is the heavier isotope, and organisms will select against it in their tissue. And it actually increases as you go up the trophic levels in a predictable way. Empirical evidence tells us that 3.4 per thousand % or per thousand enrichment with each trophic level happens with nitrogen 15. So you can actually calculate a fish's trophic position if you have information about the nitrogen signature of the baseline as well as the nitrogen signature of the tissue. So together, these two metrics help us to understand how much an organism is relying on littoral energy, as well as where it is eating in the food chain, whether it's moved up or down and such. So we use these values in the following analysis approach to understand the effects of zebra mussels on the fish community. First, we did paired t-tests to compare baseline corrected 13 sea levels in invertebrates. Then we used the mixing models described on the previous two slides to determine and compare the proportion reliance on littoral energy in fish as measured by the 13C concentrations, as well as the trophic level of fish as measured by the 15N concentrations. And finally, we used a Bayesian approach called sample size corrected standard ellipse area to estimate isotopic niche area for each species. We used these then to assess differences in community niche structure by comparing ellipse size and positions and whether or not they overlap with other fish species, and to estimate the trophic range of littoral sea reliance, and the standard corrected ellipse area for the entire fish community in each lake. So our first clue as to what's going on in lakes with infested with zebra mussels comes actually right away in the invertebrates. So I'll just orient you here to this figure a little bit. Remember, Lake Ida is the lake that up until the very end of our study did not have zebra mussels. Lake Carlos was infested several years before. Lake Carlos is those open circles and Lake Ida is the dark. On the left axis, or the vertical axis, we have baseline corrected 13C, and that dotted line at the 0 indicates almost total reliance on pelagic energy. So the 0 is pelagic energy reliance, and the higher you go on that range, the more reliance on littoral energy. So as you can see, the species of secondary invertebrate consumers on the lower rank there, damselfly, dragonfly, and water scorpion, all showed significantly higher dependence on littoral energy in Lake Carlos than in Lake Ida. We didn't see that difference among the secondary invertebrate, invertebrate producers and primary consumers, but we did see that difference in the secondary consumers, suggesting that they are eating more littoral sources in Lake Carlos. Now for the fish. These are the results of the mixing models that I talked about before. So very similar to before, on the left vertical axis is the proportion littoral energy, and the dotted line at 0.5 indicates a 50-50 reliance on pelagic and littoral. So it means the fish is eating from a diverse range of sources in the lake. As you can see, 9 of our fish species showed a higher reliance on littoral energy in Lake Carlos. But all these fish species showed no relationship between fish length and dependence on littoral energy. But almost every single one of our fish examined showed that in Lake Carlos, with the open circles where the zebra mussels were present, they're eating more in the littoral zone and relying more on littoral energy. And this is true of several different guilds of fish, including planktivores such as the blunt-nosed minnow, but also piscivores such as the walleye, largemouth bass, northern pike, etc. We also examined the relationship between fish length and dependence on littoral energy. 2 species did show an increase in reliance on littoral energy as they got bigger, the yellow perch and the rock bass. Yellow perch also showed a difference in lakes. The lake was a significant predictor with the same pattern before, Lake Carlos, having a greater reliance on littoral energy than Lake Ida. The rock bass is the one odd one out. It did not show a difference between the lakes, but it did show a relationship between fish length and dependence on littoral energy. Next, we used the N15 mixing models to understand where in the food chain the fish were eating. So similar to before, we have the trophic position along the y-axis, and the dark circles are Lake Ida, the open circles are Lake Carlos. So several species, including bluegill, blunt-nosed minnow, and walleye, showed no change in the trophic position between lakes, but largemouth bass and crappie did have higher trophic positions in Lake Carlos. So they're eating higher in the food chain in the infested lake than they are in the uninfested lake. Several fish species also showed a relationship with trophic position. So we have northern pike, smallmouth bass, yellow bullhead, and yellow perch. And they all showed a significant difference between lakes, as well as a relationship with fish length. Unsurprisingly, as the fish get larger, they're eating higher in the food chain, and therefore their trophic position increases. However, for pumpkin seed, there was no significant difference between lakes, although there was a relationship with length. And again, with rock bass, as similar to the reliance on littoral energy, there was no difference between lakes, but there was an increase in trophic position as the fish length got longer. So this is the results of the standard ellipse area that I'm going to take just a couple seconds to explain so you can orient yourself on this image. This image is a really great way to visualize what's going on in the food web. And on the left axis, the vertical axis, we have trophic position above primary consumers. So the higher you are along that axis, the higher you are in the food chain. Along the lower axis is the proportion littoral energy. So the further right you are, the more reliant you are on littoral energy. Lake Carlos is in the solid lines and Lake Ida is in the dashed lines. And almost right away, without even doing any statistics, you can visualize what's going on. Most of the fish species are shifting up and to the right in the infested lake. And what this tells us is that perhaps they're moving to more reliance on littoral energy. You can see this is true especially for the piscivores. A lot of them are moving up in the food chain and over as the in the infested lake as opposed to the uninfested lake. And if you were to draw a line kind of in the middle of each of those clusters, you could see that Lake Ida was clustered right around 40% reliance, maybe 50% reliance on littoral energy, while Lake Carlos would be more around 80 to up to 90% reliance on littoral energy in some species. The net result is that fish at the highest trophic levels are integrating more diverse energy sources in Lake Ida than in Lake Carlos. Recent research has suggested that larger or higher in the food chain fish are able to integrate diverse energy sources from different areas in the lake. But in Lake Carlos, our fish are eating at a higher trophic position and are eating less diverse food sources, suggesting that there might be increased competition, especially at the highest levels of the food chain. And this is illustrated in this image. There's a lot going on, so I'll explain it again. On the left axis, we have proportion littoral energy. On the right, trophic position. Open circles are Lake Carlos, dark are Lake Ida, and as you can see, there's a significant shift upwards over in the overall fish community, with more fish relying on littoral energy in Lake Carlos with the infested zebra mussels. In addition, the fish at the highest trophic levels in Lake Ida are eating right around 50 to 60, maybe a little bit higher, 70% littoral energy, but they're also integrating a diversity of resources. You can contrast this with fish at the highest trophic positions in Lake Carlos, in the far right side of that dotted line, that are relying almost entirely on littoral energy in some cases. However, we didn't find any difference in community level metrics. This graph shows the Bayesian credible intervals for each of these values. So we didn't find a difference in the trophic range in terms of the size of the food web, nor did we find a difference in the range of littoral proportion among the different fish species. Additionally, the overall niche ellipse for the entire fish community was not significantly different between the two lakes. So in summary, we found that secondary invertebrate consumers had a higher reliance on littoral energy in Lake Carlos, probably due to them feeding on more littoral prey such as amphipods rather than zooplankton in Lake Carlos. Also, 10 of 11 fish species showed a higher reliance on littoral energy in Carlos than in Ida, and those fish species also had statistically different niche ellipses, suggesting that they're feeding in a different niche than they are in Lake Ida. 7 of 11 fish species were also feeding at a higher trophic level in Lake Carlos than in Ida, and those fish at the highest trophic levels and the high predators are eating from less diverse sources in Lake Carlos than in Lake Ida. So as a result, zebra mussels could have implications for individual fish species and energy flow in the food web, but it's not clear what this means for the fish community as a whole because we didn't see differences in community-wide metrics. Predators and bigger fish may have reduced ability to integrate energy flow between pelagic and littoral food sources, suggesting possible competition and reduced productivity at the highest levels of the food chain. In addition, increasing reliance on littoral energy sources may impact more pelagic-oriented fish, such as walleye and yellow perch, that lack the adaptability to hunt in the pelagic areas, which might reduce their populations or increase competition for littoral energy sources. So, in contrast, if we're going to look into our crystal ball in the future, we don't really know what lakes will look like after zebra mussels invade. It's possible that greater light penetration due to increased water clarity could increase the primary production, especially in the hypolimnion, and as a result, the total primary production of a lake may stay the same. But if more pelagic-oriented species, such as walleye, are less able to take advantage of that littoral primary production, we could see a shift to more littoral-oriented species, such as smallmouth bass, and that might compensate and result in the same overall biomass with a significant shift in the fish community as the lake turns over to a more littoral-oriented ecosystem. All of this information is contained in our paper that came out in the Journal of Freshwater Biology last year, so I encourage you to check that out if you're interested in more. You can also always email me at my email there, and they'll also, I think, provide the emails after this. And you can also reach out to me on Twitter. So thank you very much for listening today. I'll be happy to take questions during the question time.
Thanks, Meg. We haven't had any questions submitted yet, but stay tuned and we'll see if we get some as we go, and we'll come back to you at the end.
Great, thank you.
Thanks. Our next presenter this morning is Jenilee Gobin. Dr. Gobin completed her PhD in the Environmental and Life Sciences at Trent University under the supervision of Dr. Michael Fox as faculty and Dr. Aaron Dunlop as adjunct faculty and Ontario Ministry of Natural Resources and Forestry research scientist for the Upper Great Lakes. Dr. Gobin teaches fisheries assessment and management at Trent and absolutely loves it. Over the last year, she has also worked as a fisheries research consultant for the Saugeen Ojibwe Nation, who are greatly concerned about the lake whitefish and the fishery in Lake Huron. So, Jenilee, we should be transferring presenter status to you. So if you can click the Take Over as Presenter button and then unmute your phone line, we should be all set.
All right, thanks. I think because I'm on a Mac, I have to do a screen share because that's what we established before.
Okay.
So I'm going to go ahead and do the screen share. Hopefully this all goes okay. All right, does everything look okay at your end?
Yep, it's just loading now.
Okay.
We're all set.
Awesome. All right, so thank you so much for having me. I'm really happy to participate in this webinar. And as you said, I'll be focusing mostly on work that came out of my PhD thesis that was investigating ecoevolutionary dynamics in whitefish, which the impacts of dreissenids became a huge part of with the ecological change there. So I don't have to explain to this audience the ways in which dreissenids have drastically impacted the Great Lakes. I'm sure that many of you are even more familiar with the broad impacts of dreissenids in lakes than I am, given my work focused a lot on the impacts of dreissenids on lake whitefish. So I'll talk mainly in that context today. So we do know that since dreissenids have become established in the Great Lakes, we've seen this shift in the distribution of nutrients and energy from the pelagic profundal pathways, so i.e. the offshore, to the benthic littoral pathways, i.e. the nearshore. And so with this, I'm going to turn on a laser pointer here. So with this, we have seen declines in primary production. We've seen declines in zooplankton and profundal zooplankton in the offshore. Here, in this figure, the dashed lines represent declines, whereas the solid lines represent increases. I should clarify that. But basically, we're seeing these impacts transfer up the food webs to reductions in planktivorous and benthivorous species of fishes that rely on these resources, and in turn in the piscivore communities as well. And lake whitefish are a cool water, demersal, benthivorous fish that historically occupied deep waters. And so we have seen with these changes, shifts in the distribution of whitefish in the lakes according to the depth they occupy. This figure here comes from some work that Michael Rennie has done, where he controlled for sampling effort and where we're actually looking for these fish. And these figures show the depths occupied by whitefish before dry synods with the hollow circles, and after they became established with the filled circles for the different lakes. So in A, we have Superior, where dry synods are not widespread and sort of acts as our control. And there we see this gradual decrease in the depths that are occupied by whitefish from deeper water to shallower water. In B, we have Lake Michigan. In C, we have Northern Huron. In D, we have Southern Huron. In E, we have Lake Erie. And in F, we have Lake Ontario. So we're seeing a few different things here in terms of how whitefish are shifting their depth in response to the establishment of dry synod mussels. In cases like in Michigan and in Southern Huron, we generally actually see a shift towards deeper water. But in lakes like Ontario, we see some shifting to deeper water and some shifting to shallower depths. And so a lot of the initial impacts on lake whitefish were thought to be associated with the collapse of diapraia, which is this fatty little amphipod here, and comprised a main prey item for lake whitefish historically. And after dry synods became established in the Great Lakes, we consistently saw declines in diapraia. So up here we see declines in diapraia density in Lake Huron, and then in the lower panel we see the declines in diapraia densities that have occurred in Lake Michigan. And in turn, we've also seen changes in whitefish diets. So in this figure, we're looking at the annual consumption of different prey items for whitefish of different ages, where in column A, we have the diet before dry synods, and in column B, we have the diet after dry synods became established. And this is for Lake Huron, but Pothoven and Madden saw similar results in Lake Michigan as well. And what we see is, before dry synods, we have, more dominant diapraia in the diet, whereas after dry synods, we see a shift towards the consumption of mollusks in gray. And as you would imagine, consuming this, squishy little fatty amphipod is probably a lot less energetically costly when you compare it to feeding on a hard-shelled mussel. So following the establishment of dry synods in several of the Great Lakes, we did see widespread declines in both growth and condition of lake whitefish. The figure on the left here is showing the juvenile growth, so growth of whitefish between ages 1 and 2 over time in Lake Huron. And the first dashed line represents the year in which dry synods became established. And then the second is a subsequent collapse of alewife when a regime shift was thought to have occurred in the lake. And basically we can see that growth of juvenile lake whitefish has declined over time, where in years later in the time series, growth was probably about half of what it was before dry synods invaded. So growth has been substantially impacted. On the right here, we have the decline of the condition of lake whitefish, so the weight once we account for a fish's length, so how fat you are given your length, over time in Lake Michigan. And again, we see that decline in condition, which could have implications for fecundity and all sorts of other things. So with this, we also saw declines in recruitment, the number of new fish that are added to these populations each year. In Lake Huron on the left, we have for Lake Huron on the left, we have the catch per unit effort of age 4 whitefish. So the number of age 4 whitefish that are caught when we account for how much effort we're putting in to catch them, and how this changed over time, again, relative to the invasion of the dry synods. And earlier in this time series, while it's highly variable, we do see higher numbers of age 4 fish compared to later in this time series after dry synods became established. And similarly, in Lake Ontario, here we have young of year trawl catch per unit effort over time. And in the more recent years, these strong year classes that are thought to be important for the productivity of these populations and the fishery are largely absent. So, so there's been these changes in growth and changes in recruitment, and I'll digress a little bit here because the first chapter of my thesis was like, surprise, you're going to do this chapter and it's not what I planned for, but I think every dissertation comes with some of that, because basically I was trying to develop this evolutionary model that I'll refer to a little bit later, and I was trying to fit these relationships for how population density influences growth and influences recruitment in these populations, and I just couldn't do it. And so I had to delve deeper into why that was. And so I basically posed that question of what factors are influencing growth and recruitment in Lake Whitefish in Lake Huron. And I did this using a model selection approach. So I developed different candidate models with predictor variables like some measure of population biomass. So in the table in the top, I'm showing the results for the growth models, and in the bottom, it's the recruitment models. So in the case of growth, I'm concerned about the total population biomass, whereas in the recruitment model, I was concerned mainly about the biomass of spawning fish and how that influenced recruitment. But basically, we have a measure of population density. We have some measure of time periods or these events, so the timing of the dry synod establishment and that subsequent alewife collapse that occurred later. And then we also have a measure of temperatures as growing degree days. And so I compared different candidate models with different combinations of these predictor variables along with potential interactions between them using AIC, so Akiki's information criterion. And what this metric does is it allows us to say, okay, for the amount of variability this model explains, are the additional parameters we're adding warranted? Because it applies a penalty for every new additional parameter or predictor variable you add in a model. Because you could add predictor variables to a kingdom come and get a better fit for your model, but that model's been overfit. So we want to say, okay, is this additional parameter really explaining a substantial amount of variability in the data, so given by that R-squared value. And when I ranked these models, what I saw was that my highest ranked models, both for growth and for recruitment, were models that contained population biomass and time period as predictor variables. So what that's telling me is that, okay, population density and the time period are important for explaining the trends that we've seen in growth and recruitment over time. And the way in which these relationships changed suggests that the relationship between population density and growth and population density and recruitment are different for the different time periods. And so what we were able to conclude from this study was that the ecological changes that have taken place with the establishment of dry synods have likely reduced the carrying capacity or the amount of whitefish that the environment can in fact support. And this is going to have obvious implications for the fishery and population productivity. So that became the next surprise chapter of my thesis was, okay, how are these changes going to impact the fishery? And to address this question, I took a step back from developing my evolutionary model and started with an ecological model. And I was working with these simulation models that I used data for the southern main basin of Lake Huron to develop. And the type of model I used was individual based. So what this means is that individual fish exist in these models and they experience processes at the level of an individual. So each year in the model, individuals have an opportunity to mature based on how old you are and how large you are. You have an opportunity to grow. You can reproduce, and so recruits are added to the population each year. Individuals have an opportunity to die. from various sources of mortality. So we have a constant baseline mortality. We also included predation by sea lamprey, which is size selective, given their presence in the Great Lakes. And then you could also be harvested by a gillnet fishery. So the ecological component of this model is that it allows us to incorporate feedbacks like that density dependence. How does the population biomass influence how much you might grow? Because as you have more individuals in the population, you're going to be competing more. You may not grow as quickly. So we could account for those types of ecological feedbacks in growth and in recruitment. And so I ran these simulations where I varied the ecological conditions according to 3 different scenarios. So I had a baseline pre-dry synod scenario where both growth and recruitment were high. And then I had 2 different versions of a post-dry synod scenario. One where growth was reduced, and then one where I reduced both growth and recruitment concurrently. And then for all of these populations, I harvested them with a gillnet fishery with harvest levels ranging from nothing in my controls, so no harvest at all, to really high harvest where basically if you could be caught, you would be caught. If you were large enough to be caught in the gear and you were the minimum size for the fishery, then you were harvested. And looked at the outcomes on harvest levels and sustainability of the fishery. So when we look at what happened to growth in those simulations, so here we have growth curves where we have the mean length of the fish changing as they age, and each of the different line types, the solid line represents that baseline pre-dry synod scenario. The dashed line shows the low growth rate scenario, and then the dotted line shows the scenario where both growth rate and recruitment rate were reduced. And darker colors represent increasing levels of harvest. So the first thing we see is that in the pre-dry synod scenario, growth was higher than in either of the post-dry synod scenarios. And then as we increase harvest, in the cases of the pre-dry synod scenario and a little bit in the third scenario as well, you actually get higher growth because of that density dependence. When you have fewer fish, you can grow more because of competition or the lack thereof. So that's what happened with growth. We can use these catch curves to look at what happened to the abundance of fish of different ages. So it tells us a little bit about mortality and age-specific mortality. So on the y-axis, we have a measure of abundance, so the mean count of individuals, but on a log scale. And this is for each of the different ages and the different scenarios going from left to right. If we focus on the baseline scenario, we see at low levels of harvest, as fish get older, you get fewer of them. And that slope is determined by your base natural mortality. As we harvest, mortality will increase for the larger fish, the older fish. And that's why as the more we harvest, the more these lines go down, the abundance is decreasing in these ages because they're being harvested. But you only become susceptible to harvest at a given size. When we look at the scenario where we reduce the growth rate, basically we see there's a delay in when you can be harvested by the fishery because these fish are growing slower and they're smaller. And so we're not able to harvest nearly as much and the abundance isn't impacted as much, but you'll see this doesn't necessarily mean good things. And if we look at that last scenario, we see something a little bit different. When we reduce both growth and recruitment, not only do we see, we see a few things. So we not only see impacts at the older ages, but we also see impacts at the younger ages too, which these fish are too small and too young to be harvested by the fishery. So this is indicative of recruitment overfishing, where basically by over-harvesting the spawning stock, when we do harvest, we're reducing the number of recruits that are being produced and added to these populations each year, which is further driving down these populations. And you'll notice there's only 4 lines here because in all of the higher harvesting scenarios, these populations actually did collapse. I'll give you a little teaser from the work I did with my evolutionary model. Populations don't collapse when we allow them to evolve. But yeah, I'll let you read the paper to find out more about that. So when we take together the impacts on growth and the impacts on abundance or mortality, here's what we get. So we have in the upper panels population biomass. In the middle panel, it's showing what is the exploitable biomass. So the biomass of fish that we actually could harvest, like they're above that minimum size, and then what we actually harvest for these three scenarios. And then on the x-axis, we have as the harvest rate increases. So in the baseline scenario, as we harvest, yep, the population size goes down, but we're harvesting more. In the low growth and recruitment scenarios, population densities decline, and what we can harvest and what we're actually harvesting declines substantially compared to in the pre-dry synod scenario, to the point where, yeah, in the case when we have low growth and low recruitment, populations are collapsing and harvest is no longer sustainable. So the take home from this study was that given the changes that have occurred and the likely reductions in the carrying capacity, these stocks can't sustain the levels of harvest that they could before dry synods became established. And this is something we should be considering. So to sum up and highlight those take homes again, the ecological changes that have come with dry synods have reduced the amount of whitefish that the environment can support by reducing the carrying capacity, and consequently the stocks likely can't sustain harvest that they could previously. Here I've provided the DOI links to the two studies that I talked about today, the first two here, and then I've also included the link to the evolutionary modeling that I did, where we looked at how these ecological changes are likely to interact with evolutionary responses to harvesting or fisheries-induced evolution in these stocks, and what that evolution could contribute for sustainability. And with that, I'd like to acknowledge all of the contributors to this work that provided funding, that provided data, without whom it wouldn't have been possible. And thank you all as well for listening. Oh,
thanks, Jenilee. As a reminder, for those of you with questions, please type them into the chat box. We are starting to get some questions submitted, but we're going to go ahead and move to our last presenter and then start to answer those questions after that. So thanks, Jenilee. If you can stay with us, and we'll get to any questions that come in with you after the next presentation. So our last presenter is Dave Caroffino. Dave is a Michigan native who began his education at Lake Superior State University, where he received a bachelor's degree in fisheries and wildlife management. He then went to the other end of Lake Superior and pursued a master's degree at the University of Minnesota, conducting fisheries research in the Duluth area. He rounded out his education by earning a PhD in fisheries from the University of Alaska Fairbanks. For the last 11 years, Dave has been the Great Lakes fisheries biologist for the Tribal Coordination Unit within the Michigan Department of Natural Resources Fisheries Division. He's involved with cooperative fisheries management with federal and tribal partners as they work together to implement the terms of the 2000 consent decree, something about which he will explain more during his talk. And so, Dave, we are handing off presenter status to you.
We're having a bit of a technical difficulty getting your presentation loaded, Dave, in that my computer is frozen. Oh, here we go. Okay, sorry for the delay there.
All right, just give us a minute. We're transferring presenter status to you, and then we'll get your phone unmuted, and we should be ready to go. All right, we can see your presentation. Dave, you may have to unmute your microphone.
did I have?
Dave, we're just trying to locate your phone line. If you could type your the area code of your, maybe the first 6 digits of your phone line into the chat box, then we can quickly locate you and help get you off mute. Apologies, everyone. Will you bear with us while we work through this audio challenge? Hi, Dave. If you could try to hit *9 on your telephone. If the *9 doesn't work, you may want to try hanging up your phone and dialing back in. Oh, it looks like we may have got your line unmuted.
Can you hear me now?
Yes, we can.
Not quite sure what happened
there. Sorry about that.
Okay, so are we all set? I think
fun.
I'm on presenter mode here, so you can see the title slide and you can hear me, so we're good to go?
Yes, we are. Please proceed.
Okay, yeah, sorry for that delay there. That was kind of strange. I appreciate the opportunity to share today with some of the work that we do here within DNR and quite a few of our partners. It was great to hear Jenilee's talk right before this, because there's going to be a few parallels here to some similar things, as we both kind of use whitefish here as an indicator of a lot of stuff that's going on. And one of the things that scientists like to do is we like to look for cause and effect relationships, and sometimes it's clear and evident and things are easy, like lightning causes thunder, but ecology isn't always that simple. Those direct cause and effect relationships, they exist, but they're very difficult to measure because of the complexity of the environment. So in this case, we can make observations and inferences that clearly support the idea that the ecosystem changes sparked by invasive mussels have certainly at least contributed to fishery level changes that have impacted people and their livelihoods. So my plan for today is to provide you with an overview of a key fisheries management structure that exists in the Great Lakes, and kind of share how that's been impacted by changing fish populations, and a little bit about the role of how invasive mussels have played into that. So you can see here, obviously, whitefish on the title slide. You're going to see that illustration show up here in a number of slides as we get going, because whitefish have been and continue to be, despite their lower population numbers, the most important commercial fish species in the Great Lakes. So as I get going here, I have to acknowledge right from the start that a lot of the data and things that I'm going to share today are not all Michigan DNR based. So they come from partnerships that we have, and you're going to see quite a few graphs, and most of them are simple, I promise, that illustrate population metrics and trends. And the data for those have come from 5 different tribal governments listed on that top row, our federal partners, Ontario, and then the group that kind of makes sausage out of all this stuff is the modeling subcommittee. The MSC is a group established by the 2000 consent decree, and it's made up of biologists from 5 different tribes, the state of Michigan, and the U.S. Fish and Wildlife Service. Kind of full disclosure, I've been co-chair of that group for the past 10 years, so the good, the bad, and the ugly, I've kind of seen quite a bit of it. So to give you a brief idea of the management structure that's in place, I really have to go back and give a history lesson. All of the state of Michigan is covered by treaties, and 2 of those, the ones listed in white here, are treaty areas where the adjacent waters of the Great Lakes are actively fished by tribal members. The Treaty of La Pointe is the 1842 treaty area in the western part of Lake Superior. And then the Treaty of Washington, often referred to as the 1836 treaty area, covers the area east of Marquette in Lake Superior. In Lake Huron, it's from Alpena northward, and then from Grand Haven, really all the way to Escanaba in Lake Michigan. So my focus here for today is going to be to talk about the Great Lakes waters adjacent to the 1836 treaty area. In the 1836 treaty, the tribes reserved the right to fish in the Great Lakes. And after Michigan became a state in 1837 and started regulating commercial fisheries, those regulations were really liberal, and many tribal fishers actually had state licenses. After the state introduced Pacific salmon, the focus really shifted from commercial fishing to recreational fishing. Gill nets were banned in the commercial fishery, and after that ban was put in place, in 1971, a tribal member challenged that regulation in court, which sparked really more than a decade of uncertainty and legal battles. Eventually, a federal court, when that ruling was later upheld on appeal, that ruling was favorable to the tribal interpretation of treaty language, and Michigan could not regulate tribal fishing because doing so would infringe upon a treaty-protected right, which in the U.S. supersedes state law. So that period from the initial citation and when the court battles really started in 1971 up until an eventual settlement in 1985 was a period of severe unrest and social conflict as people were trying to kind of figure out what this new era was going to be about. It was a changing fishery. Eventually, the state, tribes, and the feds all kind of came together and negotiated a way that everybody could share and co-manage the fishery resources within the 1836 treaty area in the Great Lakes. That was called the 1985 Consent Decree, which had a 15-year lifespan. So after that agreement was nearing its conclusion, the parties, which in this case now were the 5 tribes you saw in the acknowledgement slide, along with the state and the U.S. federal government, came together to negotiate the 2000 Consent Decree. That's the agreement under which we currently operate. It has the backing of the federal court, and it seeks to provide fishing opportunities for all parties and ensure that populations are managed in a sustainable way. So the 2000 Decree has at its backbone a management structure that includes annual harvest limits calculated based on target mortality rates that are supposed to be sustainable. So fisheries have changed dramatically during the course of the 2000 Decree. Of course, as the lakes have cleared up, nutrients are sequestered on the bottom, light penetration has increased. It's really impacted catchability of fishing nets, but it has allowed some species to thrive, such as Cladophora. The photo here you've got on your screen, the bottom of that boat was not painted green at the factory. That's Cladophora coating the bottom as it covers gill nets that are coming in. So there's a lot of impacts to fisheries, not just on the ecological end, but just the practical aspect of fishing has changed as well with changes in the lakes. So where I'm going to go now is I'm going to show you a series of population trends through graphics, and I'm going to start with Lake Superior, because this is really kind of, as Jenilee said, this is kind of like our standard because we don't have the mussel populations there that we do in the lower lakes. I'm going to show you plots related to yield, recruitment, average age in the fishery, growth, and mortality. All of these things tell a part of the story. And after I show you Lake Superior, I'm going to show you similar graphs for the lower lakes, just kind of for comparison. So first off, yield. Yield on its own can be really deceptive because commercial fleet size, individual effort, abundance of target and incidental species, market forces, all kinds of things can drive yield. But the intent isn't to really kind of show all these ups and downs, but the intent is to show that, kind of in our modern era, in recent decades, we're basically near kind of our peak harvest of whitefish, at least over the past 100 years in Lake Superior. And I'm going to also kind of zoom in and show you like a case study or a representative management unit, so to speak, that has a similar trend to what we see across the lake. And in this case, I picked Whitefish Bay. Whitefish Bay is in the eastern end of Lake Superior. It's fished very heavily by tribal entities. They're the only folks that fish there. There's no state commercial fishery for whitefish there. But yield in this unit over the past 30 years, which is the time period over which we modeled detailed population dynamics of these stocks, really over the past 2 decades, we've seen a lot of stability in the yield. There's a bit of a higher yield you saw in the late 1980s. That's largely driven by some higher recruit classes, which I'll show you in a second here. But this is representative of most of the stocks in Lake Superior, and it's really a kind of a model of stability. If we jump forward and look at recruitment, this plot is the estimated number of age 4 fish in a given year. Again, this is for Whitefish Bay. This is kind of our case study unit. Age 4 is the first age that we model in our population assessments. And you see some ups and downs here, of course, which is what you would fully expect as you examine recruitment, but there's really no trend in this data over time. There were a couple strong year classes in the early mid-1980s that produced some of those higher yields you just saw, but basically over a period of 20 plus, almost 30 years, we've had recruitment kind of locked in between 200,000 and 400,000 fish, which is not common. That's remarkably stable to see that. Those recruit data come from our assessment models, and dealing a lot with management agency and constituents and fishers, sometimes there's some skepticism about models. People don't like model output. So there's other ways to look at similar things. A more basic means of explaining population structure could come from just looking at raw data. So we sample commercial fisheries at every major port in 1836 treaty waters, and this plot that you've got in front of you is average age of a fish sampled from the commercial fishery. So note that this really basically reflects and shows the same story as the recruitment graphic. There's really no trend here. Small fluctuations are expected just due to variation in sampling and aging and small variability that we did see in recruitment. But the bottom line is looking at just simply the raw data we collect, even as opposed to the model output, shows that Lake Superior has been remarkably stable over an extended period of time. Jenilee had mentioned growth, and I'm going to mention growth again. There's been a lot of talk about that over the recent decades for whitefish. We have seen some declines kind of in our standard here in Lake Superior, right? This again focuses on Whitefish Bay. This is average weight of an age 6 whitefish over the past 30 plus years. So we have seen some declines. Average weight went from about 2.7 pounds down to about 2.3 or 2.4 pounds. But that's nowhere close to what you're going to see here coming up for the lower lakes. Lastly, I want to show you mortality, and the reason I show this is to put it into perspective with our management targets. As I mentioned, the Consent Decree has annual harvest limits calculated based on mortality targets. For whitefish, our mortality target is 65%. That's represented by the horizontal line on this graph. And this graph has things in an instantaneous scale, so that the numbers aren't exactly the same, but that horizontal line represents 65%. Each of these individual columns is the total estimated mortality rate for this whitefish stock in Whitefish Bay. It's partitioned by source. You can see, of course, we estimate natural mortality to be constant. It still remains largely an unknown. And then we partition the fishing mortality into the different sources, trap net versus gill net. But in Lake Superior, we have different mortality rates based on where you are. Whitefish Bay, where we see right in front of us, has had some higher rates, but they're all below our target. In the modern era, rates have crept up to about 55%. But if I showed you the same graph for the whitefish stock near Marquette or near Munising, you'd see mortality rates in the 30s, 30%, 35%, much lower than what we see on the eastern end of Lake Superior. So recall, though, that recruitment in this unit and really across the period has been largely stable. And that holds true across all of our units. And mortality is well below our targets. So I didn't show you estimates of population size or biomass, but those have largely been stable as well. And it's not increasing as you might think would be possible given mortality below what we consider to be our target. So that's certainly something that has not gone unnoticed in the context of our management target. We'll talk about that again here as we jump down to the lower lakes. So the story in Lake Huron and Lake Michigan is similar, and it's very different than what you see in Lake Superior. So I'm not going to show you every single one of the graphs I just did for Lake Superior for both Huron and Michigan, because there's kind of a similar story, but I want to start with yield. So yield for whitefish in Lake Huron over time, there's a lot of things that can influence it, but the peak basically over the past 100 years was in the mid-late 1990s. We've seen pretty large-scale declines since then. This graph right here on the left side stops in 2015, but if we zoom in now to a case study unit and look at just the northern part of Lake Huron, in this unit that I'm showing here basically goes from Drummond Island westward to St. Ignace, and then south down to about Presqu'île. So it's a fairly large unit in the north part of Lake Huron in U.S. waters. This is, again, a tribal fishery unit. There's no state commercial fishing in this unit. Yield has seen dramatic declines from a peak of about 2.4 million pounds in 1995 down to about 200,000 pounds in 2018. Those declines are driven by reductions in recruitment. So these are model estimates of the number of age 4 fish in each year. So if you want to think about years they were produced, it's the year this point corresponds with -4. So a bottom out number, or the lowest number we see in 2011, those were fish actually produced in 2007. But we had peak recruitment of about 3.5 million fish, and that declined down to about 500,000 fish. So it's not surprising why yield declined the way that it did. And at this point, I have to kind of note that there are at times scaling issues with the output from some of the models that we use. So I don't have time, and there's probably not interest among most of you to really kind of go into the nuances of why that is. But what that means is I can't say with super high level of confidence that the peak recruitment actually was 3.5 million fish. It could have been 2 million fish. It could have been 5 million fish. Just like the bottom or that valley there at 500,000, it could have been 50,000. It could have been 1 million. So while we can't necessarily judge the scale with certainty, we can say that the decline and the slope of the decline, we have very high confidence in those, and that's kind of the point. The good news silver lining here, I guess, so to speak, is that recruitment does appear to have bottomed out in Lake Huron. And we have seen a few stronger year classes in recent years, although it's certainly nowhere close to what we saw to the pre-invasion period. The average age of a harvested fish in Lake Huron has steadily increased since the mid-1990s. This supports the notion of declining recruitment. As the number of young fish in our samples decline, the average age logically increases. Average age went from about age 6 up to age 10. In a few years, it was as high as 12 or 13. I think that's nuanced a bit due to some sampling variability. But the past few years, we have seen a slight shift downward again, and that's produced from some higher catches of young fish, but also fewer samples overall as the fishery has contracted. If we jump over to Lake Michigan, in the kind of our long-term look at yield, we see similar things to Lake Huron. Our peak was in the late 90s, but the one difference is we do see a kind of a secondary peak in 2010. That also shows up if we zoom in on kind of a case study unit here, which I picked Gobin Way, which is in the northern part of Lake Michigan. During this 2000 consent decree, yield in Gobin Way peaked in 2009 at 1.1 million pounds, and since then, just over a single decade, it's declined to 150,000 pounds, which is where it's at last year in 2018. So what's driving that? It's similar to Lake Huron. It's recruitment. We have got two peaks, though, in this recruitment graph, which is a little different than what we saw in Lake Huron. The first one in 1995, very similar to what we saw in Lake Huron. Those fish, again, were produced in 1991. Recruitment declined. It kind of fell off after that, but then it spiked back up in 2005. And it was those kind of strong year classes in the 90s and then those early 2000s that produced that secondary peak in yield and harvest that you just saw in that prior plot. The concern, though, of course, is that, similar to Lake Huron, where we've seen these big declines in recruitment in Lake Michigan, it's just been kind of a decade or so removed, and we don't appear to have yet maybe kind of hit a bottom in Lake Michigan. So a little uncertainty is if we're there or if we're going to continue to see reductions or if things may or may not rebound. The average age in Lake Michigan really mirrors that of Lake Huron, so I'm not going to show you that plot, but I want to jump in now and talk a bit about growth. Jenilee showed age 1, and this is age 6, consistent with what we saw there in Lake Superior. This is a big deal. The average weight of an age 6 whitefish, a catchable size whitefish in the commercial fishery, the declines began in the early 90s, corresponding to those very high levels of recruitment, and essentially probably record high levels of recruitment. And you expect some reductions just with general density dependence. But of course, with the perturbations that we saw in the lake at the same time and reductions in diapauria, it was kind of exacerbated. But we had an average weight of an age 6 fish peaking at 3.1 pounds in 1992, and then it declined down to 1.3 pounds in 2008. We've, of course, seen some recovery since then, which is positive, but that goes along with big reductions in abundance and overall stock size. changes in growth matter a lot when you start to think about management and managing fisheries. Commercial fishers sell fish by weight. So if fish go from a 3-pound average to a 1.5-pound average, you've got to catch twice as many fish to sell the same weight. Smaller fish, though, at the market are worth less per pound. Abundance is going down. Catchability from the initial slides, right, Caroffino, catchability is going down. Clearer water, fish can see the nets, and then when the nets get coated in green slime, as fishers refer to it, it's even more difficult to catch fish. Fish take longer to reach a desirable size, so they're in the lake longer, subjected to natural mortality, subjected to potential fishing mortality. So there's fewer larger size fish out there to catch, which can have a huge impact. So growth rates really matter for fishery. If we kind of put this, I look at mortality to kind of put this into context a bit with what we saw with Lake Superior, but again with our management targets. This really tells an interesting story because mortality is pretty low in Lake Michigan. And we do have older fish. There are whitefish that are 20-plus years old in Lake Michigan right now. But even with mortality rates that have rarely exceeded 50% since 1998, stocks are still declining in abundance. So harvest limits are annually calculated, and they're rarely, well, I could probably say actually they're never reached because fewer people are fishing, stocks are diminished, other species impact catchability of whitefish. But if our full harvest limits were taken and mortality was 65%, we would certainly see severe reductions in stock size. There doesn't appear to be enough recruitment to sustain even the lower rates of mortality that we're presently observing in both Lake Michigan and in some cases Lake Huron. We just don't have that recruitment potential, which is consistent with what Jenilee had just presented. This is just a different approach to get to the same conclusion. So that's kind of the story of whitefish, but the management impacts of a changing ecosystem don't stop with whitefish. So the consent decree wasn't built solely on lake whitefish. Arguably, it was built more on lake trout. In the decree, whitefish are largely allocated to tribal fishers. Their decline has certainly caused severe hardship. However, the state recreational fishery was largely allocated salmon. The overlap or intersection of those two fisheries is where lake trout are found. If you're a tribal fisher and you want to catch whitefish, which you do, when you set a gill net, you're going to catch lake trout. State fishers want to catch salmon, but if populations are low or you can't catch salmon and you're out trolling, you're going to catch lake trout. So whitefish are crucial, but they're not the full story. What has happened to the state salmon fishery? Soon after the decree was signed, it collapsed in Lake Huron, essentially eliminating that fishing opportunity for state fishers. The fisheries come back to some extent, but it's nothing like what it was. This plot you've got in front of you is total Chinook salmon yield by state recreational fishers across 1836 treaty waters. State yield peaked in 2006 at 3.7 million pounds. It's since then declined to about 700,000 pounds. The vast majority of that now, of course, is in Lake Michigan. So mussels have changed the entire environment, not just impacting whitefish, but impacting the dynamics of prey species, which of course impacts Chinook salmon. So lake trout populations have conversely increased. Lake Huron is showing signs of being on its way towards rehabilitation. Lake trout in Lake Michigan are also on the rise. It's still predominantly stocked fish in the north. There have been many claims by fishers that high abundances of both lake trout and whitefish are incompatible. Either they compete for habitat and lake trout drive whitefish from the usual places, or lake trout just consume a bunch of whitefish. There's not a lot of concrete evidence to support that as a cause for whitefish decline. And Lake Superior would certainly suggest the two species are compatible. However, that is a very common theme that we still hear on an annual basis from fishers. So regardless, the state recreational fishery has seen increases in yield, but it's gone from 150,000 pounds up to 500,000 pounds. So fishers lost 3 million pounds of salmon to gain 350,000 pounds of lake trout. That's not a trade-off that they're happy about. Tribal yield has really done the same thing. Tribal lake trout yield is up from 400,000 pounds. Now it's around 800,000 pounds. But after a tribal fishery loses 4 million pounds of whitefish, gaining 400,000 pounds of lake trout just simply doesn't cut it. So where do we go from here? The consent decree expires in August of 2020. We have a changed ecosystem. Maybe it's continuing to change. We have changing fisheries which leads to very complicated decisions as we begin to negotiate a new management structure for the Great Lakes. What is the future for whitefish? Are we going to see whitefish do we have a kind of a delicate balance here between recruitment and mortality? If we're in a new era of recruitment and they just can't produce the same number of fish they did before, what is sustainability? Is that going to continually change? What level of biomass can the lakes support today? Is stocking a viable strategy to try to recover whitefish populations? There's folks that are evaluating that right now. We are coming off a period in the late 90s in the lower lakes of record yields and record recruitment. So do we perhaps need to just adjust our fisheries and our expectations accordingly? And of course, what impact does climate change have? What has it played? What role has it played so far, and what role will it play into the future? There's numerous other species that warrant discussion and certainly will be discussed as consent decree parties get into negotiations. And we could spend tons of time talking about present and suspected future dynamics of all of these and how they might fit into a management regime. But really, lastly here, some people see these challenges as a big liability as we enter into negotiations for a new agreement that's really going to dictate management. Some of these challenges are viewed as a problem and a potential source of conflict between management agencies. There's certainly a history of conflict there, but my hope is that we can kind of do the opposite, and these challenges can be viewed as an opportunity to come together and find common ground. Despite the differences in philosophy and execution of fisheries, management agencies really have very similar missions, and that is to sustain and manage these fishery resources for the use and enjoyment of current and future generations. And if that's held in focus, we can hopefully work together to learn about and adapt to a changing environment in a mutually beneficial way. With that, I appreciate your attention and your attendance, and I'd be happy to take some questions.
Thanks, Dave. So that does conclude our presentations for today, and we'll move on to answering the questions and responding to any comments you have. As a reminder, you can use the chat box to submit your question or comment. We have got a couple of questions come in, and I am actually going to start with a couple of questions we have for Meg, which have come in. So if Meg and Jenilee, if you can unmute your lines to answer questions. And we'll go ahead. The first question we have for Meg is, will you look at changes in the species composition in Lake Ida as it changes due to mussel infestation?
Yeah. Are you able to hear me okay?
Yep, we're getting a bit of an echo, so you may want to mute your computer.
Sure.
If you're,
how's that? Is that okay?
Yep, that's better. Meg, can you hear us?
Sorry, I got muted there for a moment.
Go ahead.
So thank you for the question. I unfortunately am not working on the project anymore as I'm now at the U of M, but my advisor Kyle and Brian are going back to Lake Ida to look at the lake now that zebra mussels have been there for a number of years. They're going to be doing stabilized tope analysis again, sampling all the fish and invertebrates that we did before, and I believe also looking at fish community and fish species composition. So stay tuned for more on Lake Ida. It's really exciting to be able to have an actual before and after. It's rare that we get that opportunity in ecological studies, so I'm excited to see what that looks like moving forward.
Thanks. And we have a second question for you, Meg. Is there any movement data to support the hypothesis that walleye and yellow perch are not moving into the littoral zone in Carlos?
Not that I know of. I don't know of any fish telemetry data. Like I said before, I'm no longer working in this system, so I'm not necessarily up on all of the recent work. They will get more confirmation of the stabilized tope signatures changing when they do the sampling this summer, but I'm not aware of any telemetry work.
Thanks. So we don't have any other questions submitted at this time, but I'll wait just a minute or so to see if anybody types any in. But for the presenters on the phone, if any of you have questions for each other, I would encourage you to ask that, or anything you meant to mention in your presentation and didn't get a chance to or wanted to follow up on. You can do that, and we'll see if we get any more questions. Okay, so we aren't getting any other questions. Meg, Jenilee, or Dave, is there anything else you wanted to add before we wrap up?
Nothing for me.
Not that I can think of.
Thank you, David and Jenilee, for giving the population perspective from the actual fish's point of view. I appreciate that.
Great. Well, thank you again to all three of you for participating and presenting today. Very informative presentations, and thanks to all the participants. As a reminder, the webinar was recorded and will be posted on the Invasive Mussel Collaborative website in the next couple of days. And if you do have any follow-up questions or comments, you can contact the speakers or us at the Great Lakes Commission. Thank you, everyone.